So today I would like to give some insight on how a plant cell gets shaped and how the cell gets strong enough to hold that shape. But first, let me start with this nice building. So it's the Globe of Science in Geneva. And you see that it has this really nice spherical shape. And actually, when we want to give a building or a structure such a shape with a specific property, as human, what we do is that we follow a specific design. And in other words, so we have to impose an external blueprint on the builders, so they follow the design and build such a structure. But actually, at the same scale, you can find such a strong and robust structure in nature, like the Swiss stone pines. And what is amazing is actually this natural structure, they arise, but without an external blueprint. And what is even more amazing in nature is that this structure arises actually from a single cell. And this is plant zygote cells. So what we want to address is actually how plants can build such structure and how they achieve this from a single cell to an entire structure that is strong and robust. So we address that question not looking at the whole organism, but we actually simplify the system and we look at a single cell. And that actually gives us it's going to be easier to actually tweak the parameters if we work at the single cell level. So now let me give you another structure example in nature. So this is the weaver bird nest. So actually weaver bird, they lay the twigs and they do that at a scale that we can hold in our hands, right? And the birds take the twigs and lay the twigs, but it doesn't follow an external blueprint. And it's even more incredible that actually the shape is actually quite reproducible. Now looking at a smaller scale, about 4 order of magnitude smaller, so this is a plant cell, and plant cell actually they build a nest around them. So this nest is actually the plant cell wall, and here is the primary plant cell wall. So instead of twigs, the plant cell wall is composed of cellulose microfibrils. So let's have a look at how the structures are built. So we start from fibers. And the fibers are assembled to give rise to a structure, which ultimately gives rise to a shape. And the shape, when the bird is actually making the nest, is actually feeding back on the structure because the bird is constantly adjusting the twigs assembly, so it gives rise to a robust shape. But and that is to achieve a specific function, which is provide shelter to the bird, right? But what is also important for the nest is that it has good mechanical structural integrity, so it provides a good shelter which resists external environmental changes. So the mechanics is also quite important, and mechanics also depend on structure, and there is an interplay between shape and mechanics. So now all the questions we can ask about the bird nest building, we can ask about plant cell, because it's also made of fibers that are arranged around the cell. And the first question we ask is what happened to the fibers and to the organization of the fibers when we change the cell, the nest shape. Sorry. So here you have a single plant cell that we previously removed the cell wall around. And as you can see, it's a nice spherical shape. And actually, if you take any plant cell and remove the cell wall, you will get this spherical shape. And here, the fibers you can see in blue are actually the cellulose fibers that were just built after first step of regeneration, because the plant cell have this amazing ability to build a new cell wall. So those are the very first cell wall fiber. So what happens now if we let the cell grow? So the shape, it gets actually a cylindrical shape. So the question is how we can get this specific shape. So to explain that, I would like to do a small experiment, and for that, I would like to designate a volunteer in the room. So yeah, can you join? So I'm not dressed in green, but I have a green balloon. So the balloon represents the cell, and if you blow a balloon like that, you will get a nice spherical shape, okay? And now, can you blow this for me? So now what happens if I wrap around some fibers, like I'm building a nest with some tape, and as you can see, when the cell is growing, it will become more elongated, and we have this nice cylindrical shape. So in other words, to give rise to a specific shape, you need to lay the fibers in the wall in a specific way to get different kind of shape. Okay, thank you, Anwan. I think we can thank Anwan for that. So now the question is how we can have nicely organized fibers in the wall. So for that, actually, plant cells, they have an internal scaffold, which is the microtubular cytoskeleton. So in red, you have microtubules that are underneath the plasma membrane. And in the membrane, you have some green proteins that will synthesize the cell wall fibers outside of the cell. And in the movie, you can see the green protein that synthesize the fibers in the wall are actually moving along the microtubule. So the plant cell don't weave the nest randomly. Actually, the microtubule gives the scaffold for the cell wall. So now the question is, what happened to the microtubule network and to the fiber network in the cell wall when we change the shape of the nest? So to do that, we take naked cells and we put them into microwells of different geometry, and the wells are the size of one cell. So that way we can impose a control geometry to the cell. So we first look at the microtubule just before the cell wall is regenerated and 2 hours after the shape has been changed. So as you can see on these pictures, we see that the microtubule respond to the imposed shape, and actually in elongated shape, the microtubule are organized longitudinally. So now what we can do is to regenerate the cell wall when the cells are sitting in these specific geometries. And we expect that because the microtubule guides the cellulose synthesis, we expect to see the same organization for the fibers in the wall. So if we look at the fibers after 2 days of regeneration in the microwell system, I hope you can appreciate here that actually the fibers in the longitudinal shapes are actually lying transversally. So here we have a mismatch between what we expect with the organization of the microtubules and what is actually happening in the cell wall. So how can we explain this mismatch? So one hypothesis is that there is a signal inside the cell that will dictate the microtubule to change their orientation in order to become transverse. And one such signal can actually come from cell polarity. So here we look at a polarity marker in plant tissue, which is called basal. And as you can see, when we put the cells in the different geometry, basal is actually located in the more curved area of the shape, where the curvature is higher. So here we still have to investigate more whether this polarity can actually be a signal for the fiber organization, like the microtubule organization. So next, what we ask is, okay, we have the cells sitting in the wells. They regenerate their cell wall. So what's happening now if I remove the cell from the wells? So here you have a cell, and the orange is actually the outline of the cell. And now I will play the movie. And so we come with a pipette and gently blow the cell out of the well. And here's the 3D reconstruction of the cell once it's been released from the microwells. And you can see that the cell actually kind of keep its shape, at least partially. And to explain that, it has to do with the mechanical properties of the cell wall. So that's something we check next, is how to explain this maintenance of the shape. And for that, we have to investigate the mechanical properties of the cell wall. So to do so, we have a homemade device that is made of 2 microplates, and with this device we can grab a single cell, and we apply a control oscillating force, and we measure the resulting deformation of the sample, and from that we can actually compute the elastic stiffness of the single cell. So we did that for a naked cell that is regenerating its wall, and at different time points during the regeneration process, we probe the mechanical properties of that cell. And here what you can see is the elastic stiffness as a function of days of regeneration. And what we see is that the stiffness increases by 2 orders of magnitude. So the cells are getting stiffer and stiffer as the wall is being synthesized. So the next question we address is whether we can actually design a nest from scratch at the same scale as the single cell scale. So for that, we take some micro beads as a template, and we adsorb cell wall components at the surface of the micro beads. So the 2 nice things with this system is that we can control the number of layers of cell wall components we adsorb at the surface, and we can vary the number of layers. And the other thing we can control is the components we have in the coating. And here we choose two major components of the cell wall, which are the cellulose fibers and pectin. So what we do next is that we dissolve the internal core, and what remains is actually a shell composed of the 2 major components of the cell wall, which is here the pectin and the cellulose. So the question was then, okay, so we have this nest that we can build from scratch, but does it match the mechanical properties of actual plant cell wall? So we do a direct comparison with the same device, and what we see here is that the stiffness of the synthetic walls actually increases as the number of layers increases, and a synthetic wall with 4 bilayer is actually enough to match the mechanical properties of a 5-days-old regenerated cell wall. So here I would like to emphasize that we only put 2 components of the cell wall. So it seems that we match the same mechanical properties with a rather simple system with only 2 components. So in the end, what do we have? So I think I've shown you that the shape can actually feedback on the fiber organization, so the cell wall fibers, but also the internal scaffold, the microtubule fibers. And the shape can also feedback on cell polarity. So now what we are trying to understand is how cell polarity itself can feedback on fiber organization. Then we have seen that the mechanical property of the nest is gradually becoming stronger to maintain the shape of the cell, and that we can build synthetic nests that match the mechanical property of an actual plant cell wall. So in the experiment I've shown, the geometry was either imposed by the microwells or held still, but during growth, actually, the shape is constantly changing. So the question is whether this loop also holds when we have an actual cell which is undergoing growth. And with that, I would like to thank my amazing HFSP team. So this team brings together biomechanics from the team in France, material science in Sweden, plant development in UK, and plant biophysics in the US. And it's been a great pleasure to work with that team, and I hope we keep going. And thanks for the HFSP committee to give us such opportunity. Thank you.